factors, right? So I mean, oh, not only microbes, but since this topic is based on microbial food spoilage, so our focus would be majorly on the microbes, how microbes they spoil our food, what are the characteristics of the spoiled food. So because that is very, very important. And even in 2019 exam, FSSAI has asked one questions from this topic. So when I come to that slide, I let you know the type of question that was asked, although it was very, very basic question. So yeah, so let's begin, right? So as I said, so microbial food spoilage can occur from many factors. It could be microbial, it could be chemical, it could be physical changes, and this renders the product to be unacceptable to the consumer. So as you all know, FSSAI's main objective is to provide safe and wholesome food to the consumers, right? So now, if such factors they grow in the food, so that food is unacceptable to the consumer. So definitely, I'm sure you won't like to eat spoiled food, right? If even, for example, if the apple, if there are few, those marks that come on the apple, even then you tend to reject sometimes, right? Although the fruit might be good for consumption, but yeah, I mean, the texture does not allow us to, it makes it unpalatable. So we tend to refuse eating such foods, right? So now coming to the next point, if you see, the microbial food spoilage is caused by microorganisms which produce enzymes and that lead to objectionable bioproducts in foods. So guys, even yesterday we read, right? When there is microbial contamination, there is release of gases, right? Various gases, there is the release of various byproducts, right? And also the food texture changes, the food composition changes, and it becomes completely unpalatable. We don't love to eat spoiled food, right? We love to eat the food that have clear texture. That is why, these food companies, and also we read in one of the class, right, where different uh, I mean, additives are added to give proper texture to the food, to proper color to the food, to proper fragrance to the food. So these are the various reasons why when the food is processed food, especially when the food is processed, so manufacturers they add certain ingredients, right, to make the food palatable and to increase their sales ultimately, right? So moving ahead, so if you see, there are these are few facts just on basic general knowledge facts. I mean, since we are reading about the microbes, you should also have some factual knowledge, right? If you see, almost 25% of the all food produced globally are lost due to microbial spoilage. Yes, that is why food industry is really concerned about it, because almost 1/4 of the food that is produced globally is spoiled by microbials alone, right? So it's a huge quantity. And apart from that, not only microbes, if you take another factors into consideration, for example, there is a lot of food wastage. We cook a lot of food and then we throw it, right? So that is another kind of food wastage. And there could be another kind of food wastage, for example, physical damage. Developed countries like Europe and America and Canada, so they have specific standards. They don't accept food which are physically damaged, right? Those have physical marks on it, or if there is, they are not properly treated foods. So I mean, there is huge amount of food damage which needs to be stopped to fill the hungry stomachs, right? So if you see here, 25% of food is destroyed by the microbial spoilage alone. And that is why the food industry, they are continuously, doing the research how to protect these foods, how to reformulate these products, how to add the natural preservatives. So we'll learn about preservatives in the coming videos, because that's the reason why even chemical preservatives are added to the food. Although chemical preservatives are not good for our health, right, if you consume it on a regular basis, but this is the necessity. If they don't use the preservatives, then the shelf life of the food becomes very, very less. Then it becomes the ready to eat, right? Food becomes that food is for immediate consumption. But since the food is packaged, food is then kept in shops for selling, then food is brought to our house, we buy the food from the market, even then we keep that food for almost like, say, a month or two or three, right? So the role of uh, preservatives is very, very important. And also scientists are coming up with lot of techniques, latest techniques, which helps in food preservation. So guys, today we'll discuss about the characteristics of microbial food spoilage. So what are the characteristics when the food gets spoiled? I mean, you would be able to relate it, because definitely all of you would have seen or you would have come across this spoiled food, right? So you would see these are the basic things. Again, a very basic thing you would relate to it when we come to those slides, right? So major spoilage microorganisms. We'll also talk about microorganisms, and also that part C, detection and monitoring. So this, as I said, we'll cover it in the coming uh, classes, because the fourth unit is completely focused on the detection and monitoring. So in that we're talking about ELISA, PCR, real-time PCR, Raman spectroscopy, or I mean thousands of new techniques have come, right, which increases the efficiency, and also it helps to take the immediate action whenever food is spoiled. So if you need to take a immediate action, that can be taken, right? So we'll discuss this in unit 4. So now coming to the microbial food spoilage overview. So first of all, I'll give you the overview, then we'll move on to the characteristics. So if you see, the bacteria are responsible for rapid and evident spoilage events of proteinaceous food like meat, poultry, fish, shellfish, milk, and some dairy products. So, guys, bacteria, if you see, as you all know, bacteria requires nutrients, right? Bacteria requires water, bacteria requires optimum temperature, and bacteria requires certain pH to work, because bacteria cannot survive in a low pH, like acidic pH. So bacteria majorly they require normal pH, right? I mean optimum pH for the growth. It should be neither acidic nor basic, right? So if you see, bacteria spoils such kind of food which are highly nutritious. These are basically known as high risk foods. If you see meat, poultry, fish, shellfish, milk and dairy products, whereas yeast and mold. So guys, yeast and molds are very notorious. Why? Because these are the guys who can survive in harsh conditions. They can survive in the low pH, even they can survive in the high salt concentration. Yeah, even if there is high sugar concentrations, they can survive in those. Even they can survive in dry foods. I mean, bacteria requires water for their survival, but this yeast and mold, they are so notorious that they even don't require the water for their survival. Means water is the basic unit of life, right? So they can even withstand such conditions without water and mold. So that is why yeast and mold, they are one of the major concern for the food industry, how to protect the food from the yeast and mold, right? So moving ahead, now, guys, these are the few examples which I would like to give. These are just few examples. We will read about more of these examples when we talk about food preservative topic, because in food preservation, we'll focus on each and every food, the each and every microbes that is found in food, and what kind of the preservatives are used in those foods. So I'll give you the live examples as well, the products that you consume every day from the market. So in that will cover from food preservative topic. So if you see the major foods which are spoiled by bacteria, these are, as you all know, right? Bacteria, they requires water, proper water for their survival. Even they require nutrients for their survival, right? Even they require certain optimum pH for their survival. So they can attack majorly dairy products, even fresh fruits and vegetables. If you see it, these are fruits, these are vegetables, even meat products are highly nutritious. So bacteria are very susceptible to these foods. And even in soft drinks. So soft drinks, guys, the soft drinks which has neutral pH, because majority of the soft drinks, they have acidic pH, right? But bacteria, as you all know, they cannot survive in acidic pH. So the soft drinks which have neutral pH or basic pH, so bacteria can survive in this and can spoil such foods, right? So now moving to the foods spoiled by yeast and mold. So yeast and mold, I mean, a variety of foods, they can spoil variety of foods. So because they can withstand harsh conditions, right? So they can spoil fresh fruits, vegetables, canned acidic foods, then wheat, bread, cereals, milk, and ground nuts, pasta, jam, soft drinks, meat. So I mean, yeast and mold are very, very, very dangerous, right? So that is why food industry is coming up with new technology, new preservation techniques, new packaging techniques. So I mean, this is just to protect the food from yeast and mold, especially, and also from the bacteria, right? So moving ahead, so now guys, let's talk about the characteristic of the microbial food spoilage. So now you will be able to relate to it because you would have experienced in your daily life that whenever there is food spoilage, either is texture changes, right? Or the smell changes, right? Or its composition changes. So we will see it in detail. So let's move. So if you see the major factor, that the first thing that you see is the appearance of food changes, right? So if you see the first point is appearance. When the food looks good, then what, sorry, food looks bad, then what does it mean? So majority of the time there is a microbial growth and the mycelia or the colonies are visible on the surface. So you would have seen, right? So whenever you take the food, so there would be small microbial colonies. If you see, these are small patches, right? So these could be due to multiple factors, but bacteria are one of the major. So you could easily see the colonies, especially not on banana. If you see any liquid product, right? So on liquid product, you can see a slimy layer, those full of microbes. So it shows that microbes have formed their colonies on the surface of the food product, and it develops cloudiness. Also, guys, you would have seen if you take any take example of you say normal juice, right? So you would see the juice becomes very, very cloudy, so it is not as clear as you would have purchased it from the market, right? That is because the colonies get formed on the surface, and also the cloudiness it increases. Why? Because bacteria starts degrading the nutrients which are present in the food, and also their population increases. So that is why it is no more clear. So now the cloudiness of the liquid increases, and definitely by just looking at by first glance, you can see that now this food product has been spoiled, and our brain indicates that you should not consume such food, right? So these are the examples you would have seen in your daily life. Then the B point, change in the food color due to heme or chlorophyll breakdown. Yes. So if you see the example, this is the major example. There's a change in food color. Again, if the food color is not, uh, you know, uh, up to the mark, I mean, it becomes unpalatable. Your sensory nerves, they give you the indication. First of all, whenever you see, your eyes give the indication that no, this is not the right food. I don't want to have this food, right? So yeah, I mean, these are the major changes in the appearance. So whenever you, next time if you see such foods, so just try to recall these topics, right? What happens here, why did this happen? So you need to recall all this so that you can relate to it. So that is how you should prepare for this exam, right? So moving ahead, after the appearance, the second major thing you would have seen is textural changes. That is the feel. So guys, this is the question which was asked by the FSSAI. That is slime formation. The question was very, very basic. So the question was, if there is a microbial growth in the food, what is the thing that you observe, right? So slime formation, the option, there was an option for slime formation. So slime formation is something which you see. Slime formation is nothing but again the uh, film layer, right? The film that forms on the surface. And also, if next time whenever you see the spoiled food, just do one thing: just touch that food between your thumb and the finger, and just try to rub it between your finger. You would see it's a slimy layer. It's a slimy layer, right? So this is the question which was asked by the FSSAI. So slime layer formation, it occurs. It's one of the major textural changes, and that is due to primary accumulation of microbial cells. Microbial cells, yeah, that is what FSSAI asked, right? So microbial cells. It shows that microbes have grown on the surface, so their colonies have been formed. So those colonies are responsible for the slimy layer that is formed on the text on the surface, right? And also this is due to tissue degradation, since as you all know, microbes, they keep eating the food, right? So when they degrade the food, so these are the major reasons why there's a slimy layer formation. And the second part is tissue softening due to enzymatic degradation. Yeah, I mean, this is the very, very basic example. You would have seen, for example, if you just try it once, you keep banana for like a week, don't consume it, just keep it in one corner of the house. So you would see after 5 days, the banana becomes so soft, like it becomes almost unpalatable. You would not like to have such banana. So that is because of the enzymatic reactions that keeps occurring in that food, right? Because bacteria, that they would release certain enzymes. So those enzymes would go and break down the nutrients which are present in that food. So this is how the it leads to the tissue softening. So again, it's a textural change that occurs, and our body gives us sensor that no, this food need not to be consumed, right? I hope guys it's clear till now. Just give me thumbs up if things are clear. Yeah. Okay, thank you guys, thank you so much. So I mean, these are very, very basic things. So since you are preparing for food safety officer, so you just keep observing the food that you eat, right? So just keep looking at it, what happens to it, how food gets spoiled. I mean, these are very, very basic things. So now, third point, if you see change in the taste and odor, yes, definitely. I mean, if your if your mom cooks dal or any vegetable, and after 2 or 3 days, if you forget to keep it in refrigeration, or even if you keep it in refrigerator, if somehow it gets spoiled, so when you consume, even in a first bite, you get to know, right, its taste has changed, and even order changes. So majority of the time, it's a citric taste that is released, right? So that is majorly because of these compounds. So nitrogenous compounds, they develop in that food, especially ammonia, amine, sulfide, and organic acid. So just even in a single bite, you can say the food has been spoiled. So guys, these were the three major characteristics that occur when there is a food spoilage. So first was appearance, then third was texture, and then sorry, second was texture, and then third is taste and odor, right? Fine. So moving ahead, so now guys, let's talk about the major factors which affect the microbial spoilage. As I mentioned in my previous slide, that bacteria, they required certain pH, certain water quantity, right? Certain oxygen levels, so certain temperature, and also similarly for the yeast, right? So yeast requires certain temperature, pH, water activity, oxygen concentration. So we'll talk about all that in detail now. So guys, this is the very, very important portion, because here you need to remember a lot of things, because these are very factual things, and these are a majority of the times these are asked by the examiner. So just pay attention to this, because you need to remember these figures, right? So if you see the major factors which are responsible for food spoilage, right? So it depends, food spoilage depends on food structure. It could be food composition, water activity, pH, temperature, gaseous conditions. So we'll discuss all these in detail, how these factors can impact the microbial growth, right? So moving ahead, so if you see the food structure, so guys, majorly if you see, the microbial microbes, they cannot access inside part of the food. So yesterday also, when we were talking about these microbes, so we would have seen, right, I told you that my majority of the time the fruits are sterile, vegetables are sterile from inside. Means they are not attacked by pathogens. Pathogens are majorly present on the surface of the skin of the food, right? So, but whenever they get spoiled, if there is a small cut or if there is some damage to the food during transportation, right? So that is when microbes get access inside part of the food. But normally microbes are present on the surface of the food, right? So if you see, raw food is often protected from the attack by microorganism by means of structure that are not easily degradable. Yes, so that is why the microbes, they cannot penetrate inside because of the fruit skin, right? Nut shells are very hard, so microbes cannot penetrate in that. Then there is collagen muscles, eggshell, tissues. So these all protect the microbes from entering. But whenever there is some rupture on the fruit surface, right, or whenever there is a physical damage, that is when the time when the microbes, they access inside of the food. So only microbial species with appropriate enzymes can attack these structures. Also, guys, these microbes are very specific to the food. For example, there would be certain class of microbes which are only specific for protein. That is, they would degrade only protein products. And there is certain class of microbes which could attack only carbohydrates. And there is certain class of microbes which attack only fat products. Although there can be overlap, but I mean, they are very specific. If you see, so cellulases, they majorly attack carbohydrates, right? So if you see proteases, they attack majorly the proteins, right? So yeah, I mean, they are very specific when it comes to the food spoilage. Also, the raw food prior to the harvest, it only contaminated on the surface. Yes, so raw food, as I said, before harvest only the microbes might be present on the surface only. That is for obvious reason. I mean, fruits and vegetables, they are grown either in the soil, or they are exposed to the natural environment, right? They grow in the open fields. So it's very easy for the microbes to grow on the surface, right? But after processing, so processing is the stage. I mean, when farmers, they send their product, for example, if for example, take example of apple. So apple, when farmers transport it to the food manufacturing unit, for example, juice factory, so that is where the problem occurs during transportation. If there is any physical spoilage, or when the food reaches the manufacturing unit, so during that, when the apple is crushed, when the apple is exposed, I mean, skin is removed, it's crushed. So that is the time when major microbial attack can occur. So that is why these food industries, they employ different methods to keep the environment sterile, so that this microbial attack can be minimized. But yeah, I mean, foods are most vulnerable to the spread of microbes when it's in the processing stage, right? So now, guys, if you see, now we'll talk about, like, for example, you would have, there are a variety of foods, like some are very liquidy, some are semi-solid, and some are solid foods. So if you see here, the bacteria majorly, if you see the bacteria, so they are majorly, they grow in the food which has high water ratio, right? In comparison to the other foods like semi-solid food and solid food. Why? Because these organisms can easily spread throughout the food by means of own mobility. So bacteria, they require certain water activities, some certain amount of water quantity for their growth. And also, since if the food is liquid, so they can easily move throughout the food, right? For example, if you see, if the food is solid, the movement is restricted. But if the food is solid, also you would have observed this in your daily life. For example, you keep a solid food and you keep a milk bottle, right? Just for an experiment, you would say the milk destroys faster, because now the microbes can easily access and they can easily move throughout the milk because of the liquid, right? So yeah, liquid foods are, they spoil very rapidly, then followed by semi-solid foods such as meat, stews, soup. Tinned fruits can spoil as rapidly. Yeah, I mean, similarly, the solid foods, for example, tinned fruits, for example, take the example of jam, or also the stews or the soups. So they are also uh, equally they can get spoiled as fluid foods, that is liquid foods, because at the end of the day, even stews, they have, although they are thick, but the water is high, right? So that is why these products are destroyed very at a very high pace. However, when it comes to the solid food, they tend to spoil from the outside surface inwards, this being the first surface to become contaminated. Yes, so all the solid foods, majority of them, so microbe would grow on the surface first. So from the surface, it enters inside, right? So the solid foods, it might take some time for the microbes to spoil it. But yeah, I mean, liquid and semi-solid foods, they can be easily spoiled by food, because the obvious reason is water. Because of water, they can easily move around. So now we'll talk about the food composition. So how food composition affects, right? As I said, there are certain microbes which attack only the protein foods. There are certain microbes which attack carbohydrate rich foods. There are certain foods which attack fat rich foods, right? So let's talk about food composition in detail, right? So if you talk about food composition, so nutrient composition of the food influence the growth. Yes, nutrient composition definitely affect the growth of the microbes. For example, as I said, high risk food. So high risk food are meat, fish, egg, and milk. So these are protein rich food, and they are attacked by proteolytic organisms. So proteolytic organism, there is a reason, because proteolytic microbes, they release protease. So protease can break down the large protein chain, peptide chains, into smaller proteins. So that is why proteolytic enzymes, the favorite food for them is protein food, such as high risk food like meat, fish, and eggs. And when it comes to the carbohydrates, so carbohydrate foods such as bread, flour, pasta, syrup, jams, and they are more liable to attack by fermentative organisms. So fermentative organisms, since right, uh, carbohydrates, these are majorly sugars. So sugars are further broken down into smaller sugars, and so that they can be fermented, right? So that is why the fermentative organisms, for them, the favorite food is carbohydrates like bread, flour, pasta, syrup, jams, right? And when we come to the fat rich food, so fat rich food, these are attacked by lipolytic organism, because they have lipase enzyme. So lipase enzyme, it breaks down the fatty acid chains into smaller compounds. So this gives food the rancid taste, right? Food becomes rancid, its color changes, its texture changes, so it becomes unpalatable. So these are very, very specific when it comes to the food. So food composition also plays a major role. So now food producers, producers, they have to be very, very cautious. For example, if in some food industry only protein rich foods are manufactured, right? So now they have to keep it in mind when they use the preservatives. So they know, like, their food is rich in protein, so kind of microbes that can attack this food must be controlled. So similarly, they use those specific techniques to control those microbes. So similarly, in case of carbohydrates and fats, so these food processors, they use certain techniques which could kill these microbes in food. And guys, this is really, really important topic, don't miss this. As I said, question was asked by FSSAI, although only a single question was asked, but I mean, this would form your base, because this is very, very important, right? So moving ahead, again, food composition, since we were talking about the lipids can be degraded by enzymes to produce free fatty acid. That is what I said, right? So this is again the explanation. Food becomes rancid, it gives unpleasant odor, whereas some foods have naturally occurring, yeah. So guys, again here, if you see, as we read about anti-nutritional factors, so similarly, these certain foods, they have antimicrobial properties present in them to protect themselves from microbial attack. Although the food processing industry, they add these microbes, but even naturally, these foods, they have certain microbes in them, or sorry, not microbes, I mean certain compounds in them, which help them to protect themselves from the microbes naturally. For example, if you see here, the one example that I have given is lactoferrin. Lactoferrin is present in the milk, which sequester the irons. So guys, as you know, iron, magnesium, zinc, so these are the important minerals which are essential for the microbial growth. So what this lactoferrin does is, it goes and it binds to the iron. So this iron is now not available for the microbes to consume. So if this iron is bound by the lactoferrin, so now the microbes cannot survive, because iron is very essential for their survival. So similarly, majority of the foods, they have their own mechanism to protect themselves. But But if the population of the microbes grow at very rapid rate, yesterday we read, right? So within 5 hours, billions of microbes can grow. So that is why we need to add certain chemical preservatives, because if such natural techniques, it can protect the food from the microbes if the microbes are very less. But since microbes, they grow at very rapid speed, so now we need to have to add the preservatives, or we need to have proper packaging, or we need to employ certain techniques, natural techniques, which can hinder the growth of this microbial growth, right? Fine. So now, guys, the next point is water activity. So guys, water, as I said, water is very essential for bacteria, especially microbes, right? So bacterial growth, it depends majorly on the water activity. However, yeast and fungi, they can even survive without water, because they can grow on even dry foods, right? For example, if you see seeds, you would have seen, like, if you keep the dry fruits, on your table, like for a few months, so you would see there is a growth of yeast or mold on that, right? So that is because these microbes, yeast and mold, they can easily grow even when there is no water, right? But bacteria cannot grow because bacteria requires water. So if you see water activity, this is nothing but the ratio of the vapor pressure of the food to the vapor pressure of the pure water. So you need not to go in depth what is this water activity. Just remember this: it is the ratio of the vapor pressure of the food to the vapor pressure of the pure water. That's it. Even if at all any question is asked, they might ask what is water activity, but I'm sure this question won't be asked. Even if it's asked, don't go in detail. Just remember what is it. It's just the ratio of the vapor pressure of the food to the vapor pressure of the pure water. That's it. You don't require to go in detail. So now if you see, so water activity, it plays an important role in the growth of microbes, since in the aqueous phase, the main requirement of their metabolic activity. Yes, as I said, microbes, they require water for their activity, because water is very essential. And also, if the water is present, they can grow at a rapid speed. They can, move freely into the water. So they can form colony very easily, and they can spoil the food very easily. So if you see, so guys, this is what you have to remember. So these figures, in the following slides also, I would be flashing few numbers. So this is what you have to remember from the examination point of view, and these are very important, right? So if you see, the optimum water activity for most bacteria, when I talking about microorganism, now I'm talking about bacteria, is 0.9 to 0.98, so almost 1. So 1 is the water activity that is required by this microorganism. So this is very important. Just remember these figures. So lowering the water activity inhibits the microbial growth. So have you wondered why salt is added, sugar is added, or why do we dry the food? We dry or we add high amount of salts or sugar to lower the water which is present in those foods, right? Right. So this helps, to protect the food. So just remember these figures: zero point 0.9, almost it's 1. So 0.995 to 0.980. So this is the optimum water activity which must be present. At least this must be present for bacteria to grow, right? So moving ahead, so if you see, now let's talk about the water activity of different microbes. For example, bacteria, then yeast, then mold. So guys, just remember these figures. These are very important. Again, I am saying because these can be asked in exam, and also there's a logic behind it. Why? Because for example, they can ask a question in the exam. The For example, dry fruits, right? They would say, dry fruits. In dry fruits, what kind of microorganisms can grow? Or they could say that certain food has such water activity. There, in certain food, the this is the water content or the water activity. So which microbe would attack? So you should remember, yeast grows when certain amount of water is present. So now the correct answer would be yeast. But when it comes to the bacteria, so bacteria, as I said, they require water activity greater than 0.88, right? So now you need to remember this, because question might not be asked directly. They could be a bit twisted. So be ready to answer it in any form, right? So if you see here, bacteria requires high water activity that is greater than 0.88. Therefore, it does not contribute to the spoilage of dry foods while they remain dry. So bacteria, it requires certain water quantities, so it cannot spoil dry foods. When coming to the yeast and mold, so these are very, very notorious. I mean, they can survive even at the lowest water activity. If you see 0.87 to 0.94, but some species grow in the water activity as low as zero point 0.6. So you can imagine, I mean, zero point 0.6 is nothing. I mean, there's hardly any water in that, right? So these are very, very notorious, as I mentioned. Again, when it comes to the mold, they can also grow in the lowest water activity of 0.7 to zero point 0.8. So guys, just remember these figures, because question can be asked in a twisted way, not directly. But if you are thorough with such things, amount of water activity, you can easily mention what kind of microorganism can destroy the food, right? So moving ahead, so now the next major important factor is pH. So guys, pH is also very, very important, because majority of the bacteria, they cannot grow in the acidic pH. So for example, I have shown the, a slide before this, the type of foods which bacteria can spoil. So in that, if you have seen, so the majority of those foods are basic foods, basic or neutral foods, right? Because bacteria, they cannot survive in high pH. So for example, pickles, are soft drinks which are acidic in nature. So our jams, and also bacteria cannot survive, because again, in jams we are using a lot of sugar to control the bacterial growth. Even in pickle is the classic example. So in pickle we use vinegar, right? We use vinegar. Then we also use a high amount of salt, right? So all these factors, they prevent, right? I mean, they prevent from bacteria to grow. But when it comes to the yeast and molds, so yeast and molds, these can easily grow in low pH, right? So if you see here, pH, most microbes develop around neutral pH. So as I said, neutral pH, it's around, you can say, just between 6 to 7. So this is the range for the neutral pH, right? Sorry, it mean 7 to 8, 6, 7, 8. I mean, you can consider this as a neutral range. But when it comes to the yeast, so yeast, they cannot grow, mold cannot grow. For example, there's an exception, so lactobacillus. So this is the species uh, which can even survive in the low pH. So this is an example, guys. And also ectobacter can very, very easily survive in highly acidic environment. So also remember the exceptions, but also remember the fact that bacteria, majority of them, they cannot survive in the highly acidic pH, right? So moving ahead, now let's talk about the pH of the yeast and the mold. So if you see yeast, they exhibit best growth in the pH range of 4.5 to 7. So if you see, they can grow in highly acidic to the neutral. So why food processors, they they take a lot of they keep it in mind whenever they are maintaining the pH of the food. They keep that the yeast and the mold, they are most notorious one. It provides mechanism, certain preservation techniques which can, hinder their growth. Optimal growth is optimal pH is 2 to 2.5. So you can see highly acidic. I mean, highly, highly acidic. So now bacteria, they exception. If you see, if you see this yeast, so it can even grow in pH of 5. So guys, pH is very, very important for yeast, even mold and bacteria, right? So now guys, let's talk about the temperature. So if you see, temperature also plays a major role. Why? Also, temperature is one of the technique, right? So if food is exposed to the high temperature, general practice, right? Whenever you cook food at the home, so the major why food is cooked food is cooked to destroy those microbes, right? And also to make it palatable, chewable. The major reason is to destroy the harmful microbes which are present in the food. So if you see the temperature, so temperature condition during processing, cold storage, and are one of the best, most important factor which influence the food spoilage. Right, so every type of organism able to grow within a certain temperature range, maximum growth occur at the optimum temperature. So now guys, temperature also we'll talk in our next slide. We'll talk about three type of temperature conditions, that is psychrophiles, then mesophiles, thermophiles. So let's move ahead. So you will get a better idea when we discuss them in detail. So guys, classification based on the temperature. So if you see, these microbes can be classified in threes, that is psychrophiles, mesophiles, thermophiles. So psychrophiles, if you break this word, so philes means love, right? And psychro means cold. So those bacteria which love cold temperature, they're known as psychrophiles. Mesophiles again, which require the which loves the normal temperature, that is present in our environment. For example, in Delhi, it could be 36 degree. In Mumbai, it could be 30 degree. In Southeast, again, it could be 28 to 30 degree. So normal temperature, normal atmospheric temperature is mesophilic temperature. So the bacteria, they love this temperature. And also then there are certain kind of bacteria which can survive in thermophilic temperature. I mean, they love the high temperatures. So we'll talk about this in detail. So first of all, let's talk about the psychrophiles. Again, guys, this is very, very important. Just remember this phase. It can be again a tricky question, right? So what is the range of the microbes which FSSAI can ask? The range of the microbes, even it was asked, one of the question was asked by FSSAI in the previous year. So that was purely related to the psychrophiles. So what is the range of the psychrophiles which can grow? I'm just giving you the example. I'm not sure of the exact question. So it was related somewhat to the sub-zero temperature, right? So now if you see psychrophiles, so these are the organisms which have growth range of -5 to 20 degrees. So guys, these microbes, they can survive in sub-zero temperature. I mean, below 0 degrees Celsius. If you see -5 degree to 20 degrees Celsius, right? So if you see the minimum growth, so guys, it does not mean that if the range is -5 to 20, but if you see in the next line, minimum growth occurs when the temperature is -5 to 5 degrees Celsius. I mean, although they love the lower temperature, but if you see the maximum or the optimal growth occurs at temperature 15 to 12 to 15 degree, and the maximum occurs at 50. But the thing that they want to say is, even they can survive if the temperature, if you refrigerate your food, right? Even then the psychrophiles can attack your food. So this is the example that what I'm trying to explain you here is, so this has a wide range of temperature survival. Even if your food, you keep it in the freezer, even then your food can get spoiled because of these psychrophiles, because they can even they can even survive in -5 degrees. But these are the optimal range. So but here the minimum growth would occur, because already refrigeration is a preservation technique, right? So preservation technique, it's trying to protect the food, so there would be minimum growth here. But if you see, optimum condition is 12 to 15, and their maximum growth would occur when the temperature are 15 to 20 degrees Celsius, right? So example, if you see the example, so examples are Listeria, monocytogenes, then Pseudomonas, then Yersinia. So these are few examples. You can read more about these examples so that you get a better idea about this, right? Fine. So now moving ahead, mesophiles. So if you see, it's the category in which most human and animal pathogen and food spoilage microorganisms are found. So yeah, mesophiles, majority of the microorganisms that you find in food. So we read about these microbes, right? E. coli, Salmonella, Clostridium, Staphylococcus. I gave you a chart. You remember yesterday also we discussed. So these all microbes, they fall in mesophilic category. So now guys, try to relate it, right? So now the chapters, there would be a link. So today we are reading about mesophiles. So now you remember all those bacteria which you read yesterday, they fall in mesophiles, mesophilic class, right? So it's a category of the food It's a category of the bacteria which are mostly, in human and animal pathogen. I mean, they fall in the, they can spoil the food very easily, right? Normally, the body, human temperature or animal pathogen, right? So if you see the growth range, the minimum growth range for them is 5 degree to 15 degree. Optimum is 30 to 45, and maximum is 35 to 47. Beyond 47, they cannot grow. 47 is the maximum range to which they can survive, and minimum is 5 degree. 5 degrees minimum. If the temperature are lower than 5 degree, then they cannot survive. Just remember, guys, these figures are very, very important. And examples, if you see, examples are E. coli, Salmonella, Clostridium, Staphylococcus, right? I hope, guys, it's clear and you are really enjoying the lecture, right? Just give me thumbs up if things are clear until here. And in case of any doubt, I'm always available on WhatsApp. You can WhatsApp me after the class. Just give me thumbs up if things are clear. Yeah. Okay. Fine. Thank you. Thank you for confirming. So now moving ahead. So if you say thermophiles, so guys, as the name suggests, thermophiles, right? So they love high temperature, thermo. I mean, if you see the maximum temperature, they can survive in 90 degree. I mean, 90 degree is really, really high, right? On 100 degree, water starts boiling. So these are the microbes for the minimum growth. If you see, their minimum growth only starts from 40 degree to 45. The optimum best temperature for the growth is 55 to 75, and the maximum it can grow until 90 degree. Beyond 90 they won't be able to survive, and below 40 they won't be able to survive. So guys, these are the thermophiles. So temperature plays a major role, but again, microbes also, there are several category of microbes which can, survive in different temperature ranges. So if you see Clostridium and Geobacillus, so these are the examples of the thermophiles. Again, as I said, there could be number of examples. Just remember 4 to 5. That's it. Don't just go and read all the thermophiles. Just remember 4 to 5 thermophiles, right? Also for same goes for mesophiles and psychrophiles, right? Fine. So now moving ahead, let's now talk about mold and yeast. So guys, if you see mold and yeast, so they grow best at room temperature. Yeah, I mean, these are the guys who love, who can survive anywhere. If you see, they can grow at room temperature or even below. And also there are some, therefore some assume great importance in this spoilage. That is why, even at chilled temperatures, that is why food processors, they need to take care of these two guys, yeast and mold, because they are very, very notorious to control. They can survive in low pH, they can survive in low activity, they can survive in low and normal temperature, right? So yeah, I mean, they majorly grow at room temperature. Best temperature for them is room temperature. And some yeast species like Candida, Cryptococcus, then Rhodotorula, they can grow at temperature less than 0 degree. Even they can survive at 0 degree temperatures, even in freezing conditions, right? So thus can spoil frozen food, which is, although it is quite uncommon. So these guys need to be looked after. Even yeah, in future, if you try to enter into the food field, I mean, if you try to enter the food manufacturing unit, right, you plan to open your own food business. So make sure, guys, you take care of these two guys here, molds and yeast. Bacteria, although it can be tackled, but these guys are very, very difficult to tackle, right? So moving ahead, so next point is the gaseous condition. So guys, as you all know, bacteria, it's a living organism, right? Although yeast and mold, they are also living, but they can survive in aerobic as well as anaerobic conditions. But if you see bacteria requires oxygen. So if the bacteria does not has oxygen, so it cannot survive. But if you see here, the spoilage by the aerobic organism occur at the surface of the food. As you know, the surface is the place where oxygen is present, right? So that is why you would find bacteria majorly, since they are aerobic, they are majorly present on the surface of the food so that they can survive. But when it comes to the anaerobic organisms, they can grow within the food held under anaerobic conditions, such as inside cans. So if you see, yeast can grow in both aerobic and anaerobic condition. right? But molds are also aerobic. So guys, this you need to remember: bacteria are aerobic, mold are aerobic, but yeast, it can survive both in aerobic and anaerobic condition. So can you imagine, I mean, how difficult it would have been for the food processors to ensure that you get the right quality food, you get the uh, microbe free food. That is why a lot of these preservatives are used. Because when we were talking about the chemical additives in our previous class, so a lot of students, they were doubting that why then FSSAI is allowing these chemicals. So guys, these are the reasons why FSSAI has allowed the safe usage of this chemical in the food to take care of all these spoilage organisms, all this spoilage which might take place and further, can affect our health, right? So now, guys, last, since the second part of this topic is techniques which are required for the identification of these microbes. So as I said in the beginning only, that the fourth unit, if you see your syllabus, the fourth unit is completely based on the techniques that are required for, analyzing the microbes which are present in the food. So we'll do this topic in detail when we discuss the unit 4, because there is no point in repeating. And you once we cover substantial portion, you would see there are a lot of topics which are overlapping in the FSSA syllabus. So there is no point repeating such things. So I'll just give you the brief. So what happens is, so guys, classical techniques, as these are older techniques. It takes a lot of time, and a lot of labor goes into it. A lot of lab you need a lot of complex apparatus to use in labs to identify these microbes. So these are the classic techniques. So if you see, classic microbial methods require multiple culturing steps for the microbes to be isolated and identified, right? So for identification, we need to have number of steps before we reach to a certain conclusion. So they may not also provide results quickly. So these techniques, classical older techniques, they cannot provide results quickly. However, there are recent developments, since technology is evolving every day. So now DNA sequencing, it plays a major role when it comes to identification or analysis of the microbial food microbes which are present in the food. So DNA sequencing is one of the technique. And also or the data obtained by analysis of DNA, it can tell us about the genus or the species level. I mean, we can go that depth, right? We can completely found what's the genus of these microbes, what's the species of this microbe. So with DNA sequencing, things have become very, very easy and also very, very fast. But again, these techniques are also becoming uh, classical nowadays, because now a lot of another techniques, if you see here, I have just given the example. So classical technique, for example, ELISA again takes a lot of time, lot of efforts, lot of time goes into. Since you all are science students, you would have worked in labs, right? So you would have used ELISA. It's a cumbersome task, setting up those plates and ensuring you get the proper results. And also when analyzing, so analysis also does not give proper results, it takes a lot of time. So these techniques are time consuming. ELISA, then PCR also take a lot of time. But now if you see, the lot of new techniques like real time PCR has come. Then DNA microarray, biosensor, infrared spectroscopy like Raman spectroscopy. So guys, all these techniques, principle, how they work, so we'll do all this in detail in the next unit, right? So this is just an overview which I wanted to give it to you. But yeah, in detail we'll do in next class. So guys, again this technique, it's really, really interesting thing which I came across. So now scientists, they have evolved a new method. So now the microbes, when they release certain metabolites are nothing but the certain compounds which are released by the microbes. So with the help of these compounds, it can be easily found what kind of microbes are present. So if you see, these are known as MVOCs. So microbial volatile organic compounds are generated in the process of spoilage. So whenever, as you all know, whenever the food is spoiled, so there are certain compounds which are formed, there are certain gases which are released, right? So these are known as microbial volatile organic compounds which are released. So amount of the MOVCs can reflect the condition of the microbial growth and food spoilage directly. So with the help of these metabolites which are released, so scientists, they are able to, they have generated a formula through which, or a technique through which they can easily identify. If you see the examples, so with the help of example, you would understand this better. So volatile amines, so whenever there is a amines found in the poultry meat, so this presence of these amines, they suggest that the food is spoiled. So amine is nothing but the metabolite which is released as the food is destroyed, right? So similarly, coming to the alcohol, so in alcohols, ethyl hexonate and ethyl octonate, they are found in the beef sample contaminated